Okay, now we're on to part 5 of our discussion of the digestive system, chapter 23 in our Human Anatomy and Physiology text. And we're going to talk a little bit about the small and large intestine, some anatomy and some functions of those two parts of the alimentary canal. So, digestion in the small intestine, the chyme coming down from the stomach contains partially digested nutrients, carbohydrates and proteins, and undigested fats. I guess that's a little unfair to say. We have said that there's some lipase that's secreted in the stomach and possibly in the saliva, but mainly undigested fats. So in the small intestine, we're going to have a lot of segmentation, mixing process type of motility, and it's going to allow the enzymes from the pancreas, we talked about, to complete the digestion of all the nutrients down to their monomeric subunits. We're going to absorb a lot of water. Now, there's a lot of water in our diet, but even more water is invested in the digestive process. The amount of gastric secretion may be as much as 2 liters in a meal. Half a liter of saliva production, more secretion, liters of secretion by the pancreas and so forth. So a lot of water is invested, and all that water comes from the blood, of course. And so we're going to start really reabsorbing water in the small intestine along with all the nutrients. So we'll reabsorb the nutrients, we'll reabsorb the water. We'll talk later on about the actual mechanism of that reabsorption, but just for fun to give you a preview, it's exactly the same as reabsorption of the filtrate in the proximal tubule. Primary active transport, some facilitated diffusion, secondary active transport to get all the nutrients, and then osmosis to reabsorb water, to absorb water in this case. In order to do that, we need a pace of this process. We need to deliver the chyme very slowly from the stomach down into the duodenum for a couple of reasons. We need time to digest and absorb the nutrients. We also need to make sure that we can neutralize all that stomach acid. And how do we do that? Well, we need to get bile from the liver coming into the small intestine, which will emulsify fats, allow us to get that immiscible fat that can't mix with water, break off little tiny bits at a time, essentially on the molecular level, and form some complexes, some micelles they're called, that allow the lipases to get at the fatty acids to digest them down to, or the triglycerides to digest them down to fatty acids. We need the enzymes from the pancreas. That's where all those digestive enzymes come from, as we said. We need bicarbonate ions from the pancreas to neutralize the stomach acid. And we need a lot of mixing, right? So those are the properties that are the essential ingredients for successful digestion and absorption in the small intestine. The small intestine, again, does mainly segmentation by way of motility, muscular action. A lot of just mixing back and forth, contracting, alternate places in the small intestine, and we simultaneously mix, but also slowly move the chyme down towards the colon. The ileocecal valve. So remember, the small intestine consists of the duodenum, jejunum, and ileum. So the last part is the ileum, and there's a valve at the beginning of the large intestine that's called the cecum. So the ileocecal valve controls the movement from small intestine into large intestine. What controls the actual motility is the parasympathetic neurons that interact with the enteric plexus, myenteric plexus, and also sympathetic neurons will inhibit all of the motility in the small intestine. Parasympathetic activates, sympathetic inhibits. Here's a beautiful micrograph showing the microvilli on the simple columnar epithelial cells of the intestine. On all the surface of the villi are epithelial cells, and each one of those epithelial cells have these microvilli on the surface. You can see actin filaments that give them their bristly-like stiffness, and they're covered with the cell membrane packed with transport proteins to absorb all of our nutrients. Peristalsis is the really rapid movement of materials, chyme from one place to another. Once we activate that process, we will really move a lot of stuff along through the digestive tract. The process is designed to get the last bits out. Once we finish the digestive process, we have new material coming in. We want to get all the bacteria and undigested and digested material out of the intestine so that fresh stuff can come in and start the process all over again. The duodenum, remember, is only a short segment of the intestine. We're going to periodically move the chyme right down into the ileum, the jejunum, through the jejunum and into the ileum over the course of a couple of hours of time. Waves of contraction every couple of hours to move that stuff through. But it's not just that. There are some other stimuli that are going to be involved in activating that motility, as we'll see in a second. Here's just a cartoon drawing showing peristalsis. Segmentation can mix but also move material down through the small intestine. Peristalsis is a wave of contraction that just moves stuff right along. So intestinal motility is controlled or automated by the enteric neurons, the myenteric plexus. Cholinergic neurons activate that myenteric plexus to increase, as we said, increase motility. No real extra material here that you need to worry about. So as we're continuing to move stuff down through the ileum, eventually we're going to move stuff into the large intestine. And so when we activate motility in the ileum, we simultaneously will open up the ileocecal valve periodically to let material into the cecum, the large intestine. One thing I want you to majorly remember about motility in the ileum is, besides just segmentation for long periods of time to mix and digest, when you begin to fill the stomach, filling the stomach activates reflexes in the enteric nervous system to move material from the cecum, or from the ileum into the cecum. We've got a new meal coming. We're going to start the process all over. Let's get the material that's in the ileum now out into the large intestine, make room for the new material to come in and be digested and absorbed. That's called the gastroileal reflex. Gastrin, part of the response of the stomach to food coming in, the release of gastrin from the enteroendocrine cells. Gastrin is part of that gastroileal reflex. There's a neural reflex, and gastrin is also going to help move things along from the ileum into the cecum. And every time we activate that motility, we have momentary openings of the ileocecal valve, and then we close back up again until the next wave of contraction. The large intestine, some hallmarks of the large intestine are the funny pocket-like sacs or sections of the large intestine that are produced by the muscle action of these Taenia coli, bands of longitudinal smooth muscle in the large intestine. When they contract, they bunch it up and create these sections, these little pocket-like regions in the large intestine called haustra. Haustra is a good German word, and the Germans pronounce AU as ow, haustra. Another funny feature of the large intestine, the visceral peritoneum, the wrapping of serous membrane around the large intestine, is formed into a whole bunch of fat-filled pouches called epiploic appendages. Let's take a look at a cartoon drawing of the large intestine that will show these are the major sections of the large intestine: the cecum, the colon, the rectum, and the anal canal, and the appendix, of course. But we're going to actually talk about the large intestine a little differently. We're going to name its regions according to what's in this diagram. The cecum is the first part of the large intestine. I should have color-coded that. So here's the cecum, and you can see where the ileum empties into the cecum at the ileocecal valve. Then we have the ascending colon, the transverse colon, the descending colon, this S-shaped sigmoid colon, and then the rectum, the storage area. These little fatty droplet-looking things are the epiploic appendages, and you can see the haustra, these sac-like formations in the large intestine, again created by the contraction or the tone of the Taenia coli. The cecum, the first part of the large intestine, receives the chyme through the ileocecal valve from the ileum. Then we come to the appendix, which is attached to the cecum. It's a small, essentially tube-like structure filled with lymphoid tissue. It's part of the mucosa-associated lymphoid tissue of the alimentary canal, and there's some immune function there, but it also is an interesting thing that if you purge too much of the bacteria out of the large intestine, that's where a lot of bacteria live, and they actually have a lot to do with your overall health of your body. There's a certain combination or mixture of bacteria that are all kind of competing with each other for nutrients and space in there, and as the material is constantly moving through, bacteria are dividing and multiplying, and then they get defecated out, and there's a balance that occurs, a steady state, a mixture of certain bacteria that provide a lot of services to your body, such as producing vitamins, digesting cellulose. Well, if necessary, if we lose a lot of that bacteria from the colon, it can be recolonized by bacteria that are in the appendix. Kind of an interesting thing. You also know, I'm sure, that the appendix can become inflamed, and sometimes the opening from the cecum to the colon or the appendix becomes closed off by inflammation, and then continued bacterial growth in the appendix will cause inflammation there, and it'll begin to build up in pressure, and you can have a very painful condition called appendicitis, and the appendix can actually rupture and release all its bacteria into the peritoneal space, and that's very bad. This slide just talks about the turns in the colon. The ascending colon has a right angle turn. It's called the right colic flexure. And then the transverse colon has kind of a right turn to the descending colon. That's called the left colic flexure. And then we have the descending colon, the sigmoid colon. And here you can see what I'm talking about in this diagram. You can also see it in the previous. Actually, you see it better in this diagram. Right colic flexure and left colic flexure. We do not need to worry about the naming of the different parts of the peritoneum. The peritoneum is the serous membrane pair that lines the peritoneal cavity, the abdominal cavity, and that covers all the organs therein. But wherever the serous membrane has to kind of pair up together and leap from one organ to the other, or from the wall of the abdominal cavity to the organs, there's different names for those parts of the peritoneum, and we're not going to worry too much about those. You'll hear words like omentum and mesenteries, greater and lesser omentum of the mesentery. Don't worry about that. They're just special names for parts of the peritoneum. You can see the peritoneum covers the liver, and then there's this lesser omentum, the pair of the serous membranes that then makes its way over to part of the large intestine, or the stomach, I'm sorry. That's called the lesser omentum. Then a big swath of this double serous membrane that makes its way to part of the colon to wrap it up, so that all of the parts of the GI tract are wrapped up in these nice slippery layers of the serous membrane, the peritoneum. So don't worry about that. Just realize that the abdominal cavity is lined by the so-called parietal peritoneum, shown in green, and all of the organs are lined by the visceral peritoneum, shown in light blue. And we'll just recognize that, yes, there must be a way for those membranes to get to those organs, and we won't worry about the naming of those. Once the chyme has worked its way through the large intestine, we come to the rectum. It's like a storage area, and the rectum will trap the feces there, and it will accumulate there, and as the feces accumulates there, it will start to create a signal, a desire to defecate, to get rid of all that material. But there are sphincters keeping the material in the rectum that prevent that from happening until the appropriate time. The anal canal is the passageway lined with the stratified squamous epithelium that protects it as defecation pushes the very stiff solid chyme out of the body. And then there's these two muscular sphincters that keep the chyme in place, or the feces in place, until the right time. Here's a beautiful diagram showing the internal anal sphincter. It's a smooth muscle sphincter that's under control of the autonomic nervous system. And then we have the external anal sphincter that is skeletal muscle. It's under voluntary control, so we can keep everything in the rectum until the time that we decide that defecation is appropriate, and then we can release both of those sphincters. Incidentally, there are veins that you can see in the anus called hemorrhoidal veins, and that's just the name of those veins, but sometimes those veins can become distended. If there's portal hypertension due to excess resistance in the liver, blood will back up into all the veins of the GI tract, and one of the consequences will be that there's these distended hemorrhoidal veins will become damaged and injured during defecation, and they'll cause inflammation, and then the next time defecation happens, even more damage and very painful sort of positive feedback of inflammation forming what we call hemorrhoids. What we really mean is inflamed hemorrhoidal veins will occur, and sometimes even requiring surgery to repair that. In some areas of the world where they don't have access to good health care, there may be actually a rupture in one of these veins that causes a hemorrhage. Chronic hemorrhaging from the hemorrhoidal veins can occur, causing anemias. In this country, of course, if that were to happen, someone would be very alarmed and seek assistance, and there would be some surgical repair done to try to ameliorate that. So the large intestine has some other interesting features that we won't worry too much about, but there are no circular folds as in the large or small intestine or villi. Little different arrangement of things. A simple columnar epithelium that lines the colon, but the absorption tasks are much more reduced compared to the small intestine, where we need a lot of that extra surface area. In the colon are a lot of bacteria. When we eat food, there's a lot of bacteria in it, but it's largely killed in the stomach by the acid. And as the chyme comes into the duodenum, there's a little bit of bacteria left in there, and then it begins to multiply. And it multiplies, doubles about every half an hour, so that as we continue our way down the GI tract, we get a very large amount of bacteria that's in the colon, and it does all kinds of good things for us, such as digesting or fermenting carbohydrates in the form of cellulose, things that we can't digest with our pancreatic enzymes, but the bacteria can do it for us in the colon, and they're a wonderful job they do. They also synthesize B vitamins and vitamin K. We have no metabolic capability of producing those vitamins. We eat a lot of food that's fortified with vitamins, but the bacteria in the colon can produce those vitamins for us, and then they'll be absorbed. Bacteria do form some irritating products in the colon, but they will be released into the outside world soon enough. The epithelia of the colon prevent the movement of bacteria across the membrane into the body proper. We have a lot of mucosa-associated lymphoid tissue in there, and antibodies in the mucus on the surface of the colon to prevent bacteria and so forth, and viruses and protozoa from crossing over into the body. They'll just be defecated out of the body and cause us no problem, unless there are some especially difficult, what we call pathogenic bacteria, that have some adaptations that can allow them to circumvent this protective surface on the wall of the large intestine, and they can get into the body and cause disease. And some of those include, for example, some strains of E. coli bacteria. If you ever hear about E. coli poisoning, E. coli are present in the colon all the time. They're helpful bacteria, but there are certain strains that contain genes for producing toxins that cause disease in the body. So just an example, salmonella, you've probably heard of. Certain strains of salmonella can produce toxins that allow the bacteria to infect our body and so forth. So what are the functions? A lot of little interesting tidbits about the colon, but what is it doing for us in terms of the digestive process? Well, the colon, again, is a place where the bacteria can break down a significant amount of cellulose carbohydrate. About 20% of the total amount of carbohydrate absorbed from the intestine after a meal comes from the colon through the fermentation by these bacteria. So that's pretty cool. The colon is the site of absorption of all those vitamins we just read about being produced by the colonic bacteria. and the absorption of sort of the last remaining electrolytes. You don't want to lose electrolytes. We'll let the kidneys handle the electrolyte composition of the body fluids, and we'll try to reabsorb or absorb all the electrolytes we can in the colon, as well as bicarbonate ion. I guess that's an electrolyte. We secrete a lot of bicarbonate into the intestine to neutralize stomach acid. We always do a little extra to make sure we're not causing any problems in the digestive tract, but we need to make sure we get back all of that bicarbonate ion, because as you know, every time you produce a bicarbonate ion into the intestine, you leave behind a hydrogen ion in the blood, and so we need to neutralize those hydrogen ions by reabsorbing the excess bicarbonate. In fact, if you have severe diarrhea, one of the things that will happen is you'll have a metabolic acidosis, because you'll lose that last bit of bicarbonate all the time that's supposed to be reabsorbed. And so also this large intestine is going to push the feces finally into the rectum and finally into the anus for defecation. A lot of diseases of the colon, severe inflammation can occur in the colon that causes damage to the colon that's irreparable. Colon cancers are fairly common. Sometimes these diseases necessitate removal of parts or all of the colon. Colon is not essential for life. You can still continue to digest and absorb nutrients. There are obviously some compromises in your lifestyle that will be needed, but you can live without your colon. All right, join me for part 6 next time. We'll talk a little bit more about the large intestine and digestion and absorption. We've hit upon digestion and absorption in this little small intestine, but we should mention a little more detail. What is happening with breaking down polysaccharides? What's happening with breaking down peptides and absorbing them? What happens to the fats? How do we digest those and absorb them? So